Hello, everyone, and welcome to this awesome session of Mycorrhiza, lichens, viral, and subviral particles. Students, we have already finished Unit 1. We have discussed each and every concept of Unit 1 except for this particular topic, which was left from the biological classification chapter. Okay, so today's class is all about the mycorrhizal association which is found in the nature along with the lichens, virus, and the subviral particles which are found. Okay, so I welcome you all to this wonderful channel called Let's Crack NEET UG in English. I am Aditi Gupta, your biology mentor. And before proceeding for today's class, first let me tell you something really important about Unacademy and let me introduce myself. Okay, guys. So, first of all, this is the link to our Telegram channel. You can stay connected to all the educators of this channel through this Telegram link. Okay, you will be getting all the updates of the upcoming lectures through this Telegram channel. Okay, guys. Now, coming to myself. So, my name is Aditi Gupta and I have been mentoring students for the past 6 years for their NEET UG preparation. I myself have secured an All India rank of 61 in the prestigious GATE examination and have pursued my MTech from IIT Kanpur in Biological Sciences and Bioengineering. Okay, students. So, by profession, I am a research scientist, so I am here to follow my passion for teaching. And you are well aware of the fact that there are topmost team of educators on board on this amazing platform called Unacademy. But how can you avail maximum benefit from them? So the best option is to go for Unacademy subscription. So let me first tell you how can you proceed for an Academy subscription and what are the benefits of having an Academy subscription. Okay, guys. So first of all, you need to download this awesome app, Unacademy Learning app from Play Store or iOS. Okay. After installing it on your phone, you can go to the Academy subscription platform. There are two platforms available, Plus and Unacademy Iconic. I'll be discussing the benefits of both Plus as well as Iconic platform. Okay. But before proceeding to that section, let me tell you something that this Unacademy Learning app contains some absolutely free of cost features. Do you guys know that? See, the special classes, which those are that are available on Unacademy Learning app, are absolutely free of cost, and also the quiz sessions. Okay, you can participate in multiple quiz sessions, and you can explore and learn through multiple special classes which are available on Unacademy Learning app. So go and explore and enhance your learning experience. Okay, talking about myself, so I am available daily at 11 a.m. on Unacademy Learning app. Okay, my profile link is provided in the description box. You can follow me there to get all the updates regarding my class timings. Now, coming towards the subscription part, guys. So, by subscribing to an Academy platform, you will be getting daily live classes from the topmost team of educators, and you can select your batches because there are multiple batches running. You can select your batches according to your requirement, okay, and you can track your preparation by participating in various free mock tests or Unacademy champion leagues or Unacademy. There are multiple test series which are running on the platform. Okay, guys. So coming specifically to the benefits of having Unacademy Plus subscription, you will be getting daily live classes of all the three subjects, that is physics, chemistry, and biology. Okay, along with regular doubt clearing session. So what is included here? Regular doubt clearing sessions. Okay, well, apart from that, you guys will be having the flexibility of selecting your educator. Okay, there are 50 plus educators available on Unacademy subscription platform. You can choose your educator based on your understanding, your convenience, and your compatibility. There are no such restrictions. Okay, you are flexible to choose. Okay, then you can select your own batch. Okay, as I showed you that there are multiple batches running on the platform, so you can choose your batch depending upon your requirement. Then the complete NEET UG syllabus is covered in a structured manner, in a detailed manner, along with the detailed PDF notes, and these PDF notes are provided to you directly by the educator, and these notes are the well researched study materials which are provided to you, so you don't have to worry about which notes to follow or which books to follow, because we are here to provide you all the information, right? Then you will be provided with live tests and quizzes to evaluate your performance. You'll be provided with daily rankings. Okay, so that you can check your performance from test number 1 to test number 50. Okay, and you can check your improvements. Okay, then you will be getting unlimited access to all the NEET UG content. That means you will be getting access to the video lectures, to the PDF notes, to the live tests and quizzes of the all the 50 plus educators available on the platform. So these are the benefits of the Plus platform. Now coming towards this Iconic platform, so Iconic is a brand new platform introduced by Unacademy, and it includes some out of the box features like each and every student in Iconic will be having his or her own personal coach. And your personal coach will be there throughout your NEET UG preparation journey to keep you motivated, to listen to your problems, to listen to your challenges, to prevent you from deviating from your path. Okay, so your personal coach will be there throughout your NEET UG preparation journey. Then you will be getting a study planner so that you can utilize your time to the best of your efficiency. Okay, and these study planner will be reviewed with the help of bi-weekly meetings. Then you will be provided with in-depth test analysis so that you can learn from your mistakes. You can see what mistakes you have done in a particular test, right? And then this by this test analysis features, you will be improving your problem solving skills, right? Then all the benefits of Plus platform are included here on the Iconic platform. And from time to time, you will be getting expert motivation sessions to keep you energetic, enthusiastic throughout your preparation journey. Okay, so come on, let's proceed towards the towards the subscription part. So as I told you, there are two platforms, Plus and Unacademy Iconic. In both the cases, in both the platforms, you are getting India's best educators in interactive live classes, structured courses and PDFs, live tests and quizzes. But what's new in Iconic? Personal coach, study planner, expert guidelines, and test analysis. Okay. Now, moving towards the subscription part, you can proceed for the subscription through Unacademy Learning app or visiting unacademy.com. And if you have decided to go for Unacademy Plus platform, so there are multiple subscription plans available, but I would suggest you to buy a 1 year or 2 year of subscription plan, because the longer the duration, the more advantage you will be getting in terms of in monetary terms and also in terms of education, because you will be under our guidance, under topmost team of educators for that particular duration. And 1 year or 2 year of time is required for NEET UG preparation, guys. Okay, and those students who are targeting NEET 2021, we are already in the mid of this session. So some of you who are still confused, who have still not bought the subscription, you can proceed for the 6 months of subscription plan. And the price for 6 months is rupees 28,000, for 1 year is 38,500, and for 2 year is 56,000, right? But if you use my referral code, which is AG001, you will be getting an instant 10% discount on your course fee. That means now you have to pay only rupees 25,200 for 6 months, 34,650 for 1 year, and 50,400 for 2 years. Okay. Similarly, talking about the Iconic platform. So this Iconic platform, it includes 3 subscription plans: 6 months, 1 year, and 2 year. 6 month subscription plan costs you rupees 38,000, 1 year rupees 65,000, and 2 year rupees 1 lakh. But again, if you use my referral code AG001, you will be getting an instant 10% discount. And now you have to pay only rupees 34,200 for 6 months, 58,500 for 1 year, and 90,000 for 2 year. Always compare the monthly cost which you are paying. For example, you are paying only rupees 3,750 for 2 year per month, right? And in that and only this much amount, you are getting free live classes of daily live classes of all the 3 subjects with regular doubt discussions. Okay, you can interact with your educators, you are getting personal coach, the complete NEET UG syllabus will be covered along with the PDF notes, live test series, and the in-depth test analysis, and many more features. Okay, so always go for the best. Okay, students, so come on, let's start today's session. So today we are discussing a sort of miscellaneous topics. Okay, so we will be starting with mycorrhiza. So let me first tell you what is the association. What is the association which is known as the mycorrhiza? So mycorrhiza or mycorrhizal association is the symbiotic relationship or symbiotic association between the fungus and the roots of higher plants. Okay, so what happens in case of mycorrhiza? Certain fungi, they interact with the roots of higher plants. And generally, in those plants where, well developed roots is not found, these associations are very frequent. These associations, mycorrhizal associations, are seen in case of gymnosperms, okay, angiosperms, and also in some of the pteridophytes. Okay, so also in some of the pteridophytes. Okay, So in all these plants, that means the group Tracheophyta, it has this mycorrhizal association. Okay. Now what happens? As I told you that these roots do not have a well developed structure. Generally, they lack root cap and root hairs. Okay, so with the help of the fungal association, what happens? This mycorrhiza which is formed, it helps the roots of the higher plants in efficient absorption of the nutrient. Okay, so the fungal part, it helps in the absorption of water and minerals specifically. Okay, and in return it is dependent upon the host plant, that means the roots of higher plants, for their nutrition part. Okay, now, mycorrhizal roots, the mycorrhizal association which are present in the roots, they provide resistance to the plants to certain soil-borne pathogens. That means those infectious agents which are present in the soil and which can cause damage to the plants. Okay, so mycorrhizal associations, they provide resistance to the plants against such pathogens and also to certain metal toxicity. Okay, so now we know what are mycorrhiza. It is the association, it is a symbiotic association, to be very clear, symbiotic association between the fungus and the roots of higher plants. Okay guys, now mycorrhiza is of two types, ectomycorrhiza and endomycorrhiza. Ecto is out, endo is inside, right? So I have included the images of both the type of mycorrhiza, ecto as well as endo, although the endomycorrhizal image is a more descriptive one. But the focus which I want to show you is that the ectomycorrhiza, what happened, the fungal hyphae in the case of ectomycorrhiza, they are present only to the external part of the plants, external part of the root here, as you can see. Okay, whereas in case of endomycorrhiza, the fungal hyphae, it is penetrating the cells inside till the cortical region. Okay, so the fungal hyphae is penetrating the cell even towards the cortical regions. Okay, so ectomycorrhiza, the fungal hyphae are mainly external, whereas in endomycorrhiza, they penetrate deep inside, even up to the cortex. Okay, now what happens? The fungal hyphae in the ectomycorrhizal area, what happens? They form a complex network like structure. They form a complex network like structure in the intercellular space. That means the space between the two cells. Okay, and this network is known as Hartig net. That means the Hartig net is formed in case of ectomycorrhiza. Whereas the hyphae, it grows inside, in the intercellular as well as intracellular direction and forms VAM, vesicular arbuscular mycorrhiza. It is vesicular arbuscular mycorrhiza. Okay, you can see various nodule like structure, which is, see, this is the arbuscules. Okay, so these are the arbuscules which are formed by the fungal hyphae by penetrating in the intracellular as well as intercellular regions. Okay guys, now the ectomycorrhiza is found in approximately 10 to 20% of plants only, whereas the endomycorrhizal association is observed in approximately 80 to 90% of the cell. Okay, that means majority of the plants, they contain endomycorrhizal association. Okay, talking about the fungal part. Okay, the fungal partner in case of ectomycorrhiza is majorly from the Basidiomycetes category. Okay, and talking about the fungal part in the endomycorrhizal category, the fungus comes from the Zygomycetes class, whereas in the ectomycorrhiza, it comes from the Basidiomycetes. So this is an important point. Okay guys, so these are the two types of mycorrhizal association which are found. Now there are certain common examples which are found in the nature. For example, pinus, orchids. Okay, see, in the pinus root, ectomycorrhizal association is seen, whereas in the orchid root, endomycorrhizal association is seen, and these both examples are important from examination point of view. Okay, so now the mycorrhizal association is clear to us. It is the symbiotic relationship between the fungus and the roots of higher plants, and the fungal part is helping in mineral and water absorption, and in return what happens, it is receiving the food from the higher plant. So this is how this relationship is a symbiotic relationship which is acting, it is present in the nature. It is a symbiotic relationship. Okay, moving ahead, let us now see the second component, which is the lichens. Okay, so what are the lichens? Again, another type of symbiotic relationship. But this relationship is between the algae and the fungi. Okay, the algal part of the lichen, I will be writing it over here, algae along with the fungi. So the algal part of the lichen is known as the phycobiont part, because phyco is the term we use for algae, whereas the fungal part is known as the mycobiont part. Okay, this association was also earlier known as the husband wife relationship or master slave relationship. Okay, so it is a symbiotic relationship where algae and fungi, they both combine together. Although algae forms approximately only 1 to 5% component of the lichen, whereas 95 to 99% component of the lichen is formed by the fungal part. Okay, and the fungi, and both the component have their individual roles to play. Come on, let's check the roles. So what are the properties of lichens? So see, the phycobiont means the algal part, and the mycobiont means the fungi. So we know that algae are photosynthetic in nature. That means they will be producing their own food. That means they are autotrophic in nature. So they are preparing food for themselves and also for the fungi. So fungi is dependent upon the food on algae. And fungi are heterotrophic, we know, right? Algae forms around 1 to 5% of the lichen body, whereas fungi form majority of the component, that is 95 to 99%. The algae which is present in the lichen, they are mainly from the chlorophyceae class, that means the green algae. They are mainly from the chlorophyceae. Sometimes it is also present from the cyanophyceae class. Cyanophyceae, remember, blue green algae, kingdom Monera. So sometimes cyanophycean members are also present in the lichens, but majority of times it is present from the chlorophyceae class. Similarly, talking about the mycobiont component, so the fungi is mainly present from the Ascomycetes class. Mainly from the Ascomycetes. Sometimes the fungi can be from Basidiomycetes. Sometimes it is from Basidiomycetes, right? But most of the time it is from the Ascomycetes only. Okay, guys. Now, this association, as I told you, was also known as, earlier times it was also known as husband and wife relationship, where just like husband, the fungal part was helping the algal part in the water and the mineral absorption, whereas in return the algae was preparing the food and providing it to the fungus. Okay, now what happens sometimes when the cyanobacteria is acting, or the blue green algae, BGA, is acting as the algal component in the lichens. Okay, then they are mainly involved in the process of nitrogen fixation, and this type of association is known as diazotropes. Azo is the term used for nitrogen. Okay, so diazotropes, when the lichens are involved in the process of nitrogen fixation, because the algal component is the cyanobacteria. Okay, now lichens are very sensitive to the pollution. They are never found in pollution rich regions. So they are pollution indicators, and specifically the pollution of sulfur dioxide. Okay, they are indicated to the pollution of sulfur dioxide. They are never found in the polluted area. Okay, and also lichen act as the pioneer community during the ecological succession. Lichen is the first organic matter or organism which is found in a barren area during the process of ecological succession or development of new growth forms. So lichens, and following the lichens, mosses are present after that. Okay, so lichens, what they do, lichens, they develop cracks in the mountains, and in those cracks mosses develop, and this is how the succession takes place, right? Although lichens have various uses, based on the thallus of the lichens, they are further classified also on different types. For example, the foliose lichens, the fruticose lichens, the crustose lichen. The crustose, the thallus is attached to the crust. Foliose, thallus is leaf-like. Fruticose, the thallus is more branched sort of. These are the three different types of lichens. Okay, the dhoop which we use in the puja every time, okay, or the incense sticks, they're all developed from these lichens only. Okay, so this was the lichen association. Now moving ahead towards the next miscellaneous topic, which is the viruses. Till now we know that viruses are the connecting link. Viruses are the connecting link between living and non-living. That means viruses shows the properties of both living as well as non-living. Okay. So, according to R.H. Whitaker's system of classification, viruses were given no place to the in any of the kingdom, right? So, for viruses, there are different community of their nomenclature as well as classification. Okay. Now, viruses were discovered by Ivanovsky during 1892 while he was working in the tobacco mosaic virus (TMV). It causes disease in the tobacco plant. And due to this tobacco mosaic virus, the mosaic pattern in the leaf of the tobacco plant is observed. Okay. So, what are viruses? So, these are the non cellular organisms. We know that they are not composed of cells. They are exception to the cell theory, right? Viruses are exception to the cell theory because they are not made up of cell. And that is why here it is written non-cellular organisms having crystalline structure outside the living cell. So they have a crystalline structure and they take over the machinery of the host cells to replicate themselves. Now this is something which makes them living. Okay, this is the feature which makes them living. They have the tendency to replicate themselves. Okay, so they were first discovered by Ivanovsky. Then Bejering stated contagium vivum fluidum, that means infectious living fluid. So see what happens. Bejering, he discovered that the fluid from the infected tobacco plant can be used to infect other healthy plants. Okay, the fluid from the infected tobacco plants can cause infection to other healthy plants. Okay, so that means it contains some infectious living fluid, and that is the meaning of this term contagium vivum fluidum. Okay, and Stanley observed that yes, viruses can be crystallized, and with the help of this crystallization of the viruses, we can crystallize the virus, and they can be stored for indefinite amount of time. Okay, so viruses are even smaller than bacteria. Okay, now what are viruses? So they are obligate, that means compulsory intracellular parasites, and contain either DNA or RNA as the genetic material. So usually either they contain the DNA or the RNA as the genetic material. Okay, and they are obligate intracellular parasites because they need their host for their replication process to complete. Okay, they cannot replicate on their own. Then they can pass through bacterial filters. That means this denote that they are smaller than the bacteria. Smaller than bacteria, right? And we know that they cannot live independently. They do not have any living structures like the cell organelles or the protoplast. Okay, neither do they have the molecular respiration pathway or the ATP synthesizing machinery. They don't have any of them, and they even cannot undergo this process of cell division. Okay, but they can be crystallized and they can be stored for longer duration of time through the process of crystallization, and this is how we store the virus. This was the thing which was observed for the first time by Stanley. So all these features, it relates the virus to the non-living thing, right? See, they are made, but there are certain properties of the viruses which relates them to the living organisms. Example, they are made up of organic molecules and they do contain the nucleic acid, because we know that after the crystallization of the virus and after the study of this structure of the virus, it was found that viruses contains proteins and nucleic acid. That means they are made up of organic molecule, and those organic molecule are the are characteristically found in the living organisms. So these are the properties of the viruses which relate them to the living organisms. Okay, and they can multiply inside the host. Although they cannot multiply outside the body, but they can multiply inside the host. And they can undergo mutation, that means sudden inheritable changes in the genetic material. What is mutation? Sudden inheritable changes in the genetic material. Okay, so viruses can undergo mutation and then they can definitely pass it to their progeny. Okay, and viruses does cause this infection. Okay, and they has host specificity. So these are the properties of the viruses which relate them to the living organisms. Okay, and some of the viruses are known to contain some enzymes. Example, lysozyme. They are present in the bacteriophages. We'll be studying the life cycle of the bacteriophages. There you will understand the function of this lysozyme. Then also other enzymes like integrase or the reverse transcriptase or the protease, they are also found in other viruses like the HIV. Okay, this integrates, it helps in integrating the viral DNA to the viral DNA to the host DNA. This reverse transcriptase, it helps in the synthesis of DNA molecule from a RNA template, and protease, it helps in the digestion of the protein molecules, right? So these are some of the enzymes which are found in the HIV molecule, HIV virus. Okay, now talking about the type of viruses. So on the basis of the coating or external covering, viruses are of two types. One is the non-enveloped and second is the enveloped virus. So these non-enveloped virus, they do not contain any external coat or envelope, and that is why they are also known as naked virus. Whereas the enveloped virus, they contain an external covering known as the envelope. Okay, outside the capsid. Okay, outside the capsid, they contain an external envelope, protective covering. Okay, so the naked virus or the non-enveloped virus example includes tobacco mosaic virus and bacteriophages. These two are very important examples of the viruses. Talking about the enveloped viruses, HIV virus, influenza virus, herpes virus, vaccinia virus, there are multiple examples of the enveloped virus. As you can see, there are protein spikes present on the envelopes. Then inside to it is the capsid, which is enclosing the genetic material. Genome is labeled here. Okay, so these are the two types of viruses. Now, let us understand the different components of virus. Okay, so let us start from the outermost component, that is the envelope. Okay, so this envelope, if present, first of all, it is not a compulsory structure, that is, it is not found in each and every type of viruses. If present, these envelope are composed of proteins, lipids and carbohydrates. But the protein part of the envelope is synthesized or secreted by the virus itself, secreted by the virus itself. But the lipid and the carbohydrate component is derived from the host cell membrane, and this is important point, derived from the host cell membrane. That means the envelope contains protein, lipids and carbohydrates, and out of which only the protein is secreted by the virus, it is synthesized by the virus, but the lipids and carbohydrates are derived from the host cell membrane. Okay, and various small subunits of the envelope is present, and the subunit of the envelope is known as the peplomers. So peplomer is what? Subunit of the envelope. And in those cases where envelope is absent, then they will be termed as the naked virus. Okay, so in naked virus, the outermost structure is the capsid, that is the protective coat which is made up of protein. So protein coat of virus. So capsid is made up of various small subunits called as the capsomers, and these capsomers are arranged either helically or polyhedral, okay, in various geometric shapes to form the structure of the virus, and it encloses the genetic material in the centermost region. Okay, so we have discussed the envelope, the capsid. Now let us see the genetic material. As I told you, genetic material is either DNA or RNA. Okay, so the viruses which contains DNA as genetic material, they are known as deoxyviruses or deoxyvira. Whereas the viruses which contain RNA as the genetic material, they are known as riboviruses or ribovira. Okay, the viruses which are which infects the plants are known as the phytophagini, and normally they contains single stranded RNA molecule as genetic material. Whereas the viruses which infect the animals are known as the zoophagini, and normally they contain double stranded DNA, but sometimes they do contain single stranded DNA or single stranded RNA molecule also, but majority of the times they are double stranded DNA molecule. Okay, bacteriophages, that means the viruses which are infecting the bacteria, so they contain double stranded DNA molecule. As the genetic material, but there is an exception, 5X174 bacteriophage. 5X174 bacteriophage has single stranded DNA molecule, and this is an exception in the bacteriophage category. Otherwise, all the bacteriophages has double stranded DNA molecule. Okay, so this was about the structural component of the virus. Now there are two very important examples available in the nature. First one is the tobacco mosaic virus and another one is the bacteriophage. So let us look at their structure. See, I told you the tobacco mosaic virus, they are the naked viruses, right? They do not contain the envelope. So the outermost covering is the capsid. Okay, and in the center portion is the RNA. Okay, so this capsid is made up of small units which is known as the capsomers. There are around 2130 capsomers arranged in the capsid to form the capsid molecule of the tobacco mosaic virus. Okay, talking about the bacteriophage, so it has a tadpole-like structure. It has a head, as you can see here. It is, it is the capsid which encloses the genetic material, and genetic material is double-stranded DNA, right? Then it contains the collar, and attached to the collar is the neck region. Okay, then comes the tail sheath. Okay, and then the base plate, and through the help of this base plate, various tail fibers are arranged. So this is the typical tadpole-like structure of the bacteriophage. Bacteriophage are the viruses which are infecting the bacteria. Okay, now let us understand the life cycle of these bacteriophage viruses. Okay, because from the examination point of view, the life cycle of these viruses is important also for your conceptual clarity. So the life cycle of bacteriophage is generally of two types: lytic cycle, and second one is the lysogenic cycle. Okay, so let me first explain you how does this lytic cycle is completed in case of bacteriophage. So see what happens. First of all, this phage particle or the bacteriophage, it infects or attached to the bacterial cell or its host. Upon attachment to the bacterial cell, it starts releasing the lysozyme. Okay, and the release of the lysozyme is released from the spike particles, and the lysozyme dissolves the peptidoglycan cell wall layer of the bacteria, and then the tail fibers, they contract, releasing the double-stranded DNA into the bacterial cytoplasm. That means now the penetration has taken place. The viral DNA has entered into the bacterial cytoplasm. Okay, now what happens? This viral particle, it hijacks the host machinery, and it starts synthesizing or replicating its own DNA. First of all, its own DNA is replicated. Okay, then based on the information from the DNA, there various protein components are synthesized. Various protein components which are needed for the synthesis of head, neck, tail fibers, they are synthesized. Okay, that means the biosynthesis phase is taking place here. Then, when the DNA is replicated and various different protein components are synthesized, and the assembly or the reformation of the new virus particles takes place. So, see what happens here. There are new viral particles which are get assembled inside the bacterial cell. Okay. Now, what happens? These newly formed viral particles, they, with the action of lysozyme. Okay, and this lysozyme is also known as endolysin, because it is released inside the bacterial cell, and while lysine, because it will be lysing the cell. Okay, so what happens, this viral particle, they release the lysozyme, which is also known as the endolysin, and with this, the bacterial cell lyses, and what happens, the viral particles, the newly synthesized viral particles are released in the surrounding, now looking for their next host. Okay, so this is how the lytic cycle or the virulent cycle or the virulent phage completed its life cycle. Okay, so this lytic cycle is also known as virulent phase, or the non-temperate, also known as the non-temperate phage life cycle. Okay guys, so this was about the lytic cycle of the bacteriophage. Now here comes the second type of cycle, which is the lysogenic cycle. Which is what? The lysogenic cycle. So it is generally non-virulent. It is generally what? It is generally non-virulent or temperate phage life cycle. Okay, so the initial steps are common, like the attachment and the insertion of the viral DNA is common. Okay, virus attacks to the surface of the bacterium, and it injects its genetic material into the bacterium. But now what happens? With the help of an enzyme which is known as integrase. Now with the help of this integrase enzyme, the viral DNA is integrated into the bacterial genome. The viral DNA is integrated, that means attached to the bacterial genome. Okay, now what happens? This viral DNA, it secretes a repressor protein. And because of the activity of the repressor protein, the new viruses are not formed. See, the virus genetic material becomes part of the genetic material of the bacterium. Okay, now after some time the virus genetic material removes itself and becomes active, and this is the time when the repressor which is synthesized by the viral particle is inactivated. So initially the repressor molecule is active, but once it gets inactivated or it's deactivate, okay, so what happens, the viral particle, viral DNA, it separates itself and it starts replicating, and once the viral DNA has been replicated, the protein molecules will be synthesized, and this is how the new viruses are formed. And once these new viruses assemble, okay, these particles assemble to form the new viruses, these viruses will release the endolysin again, okay, and with the release of this endolysin, the bacterial cell will burst, releasing the newly synthesized viruses into the surrounding. So this is the lysogenic cycle. So this lysogenic cycle, it converts into lytic cycle upon the deactivation of the repressor protein. Upon the deactivation of the repressor protein, the lysogenic cycle, that means it converts from being non-infectious or non-virulent to being virulent or infectious in nature. So these are the two types of life cycle which is found in case of bacteriophage. Okay, so this was about the virus. Now, let us have a quick look on the subviral particles. What are the subviral particles? See, the viroids. Viroids were discovered by Theo Diner. And specifically, viroids are smaller than the viruses, and they are, extremely low molecular weight substances, and basically they contain single-stranded RNA loops. They do not contain any protein coat. That means the capsid is not found in the viroids. They just contain the single-stranded RNA loops. Okay, and this structure is infectious in nature. It is known to infect certain plants. These viroids are known to cause diseases in plants. Okay, example: potato spindle tuber viroid. Theo Diner, he discovered viroids while studying this particular plant potato. Okay, then chrysanthemum stunt viroid, citrus exocortis viroid. Okay, so these are the viroids. That means the single-stranded RNA loops without any protein code is smaller than the virus. That means yes, this is true that viroids contains always RNA as their genetic material, and RNA is always single-stranded in nature, and capsid is absent in their structure. Okay. Now let us see the virusoids. So what are the virusoids? It was discovered by Randalls, and they contain naked nucleic acid. Again, they contain naked nucleic acid. That means the nucleic acid is not surrounded by any structure, but they also have a specific property. They need the help of a helper virus for its replication. That means they cannot replicate on their own. They cannot replicate on their own. They need the activity or they need the help of the helper virus. Therefore, they are also known as the satellite virus. Example of the virusoids are satellite tobacco necrosis virus. Satellite tobacco necrosis virus. Okay, so viroids were only the single-stranded RNA loop. Now virusoids are single-stranded RNA loops, but they also cannot replicate. Okay. Now coming towards other subviral particles like the prions. Prions are basically proteinaceous in nature. So they are proteinaceous infectious particles. Okay. So abnormally folded proteins without any nucleic acid. That means prions do not contain any nucleic acids. They are just the proteins which are now not present in their normal configuration. That means now they are abnormally folded. Okay. So the prion proteins, also referred to as PRP, they are very resistant structure. They are resistant to various acid, alkali treatment, various enzymes like nucleases, okay, various proteases also. They are resistant to multiple chemical as well as enzymatic actions. They are similar in size to that of a virus. They are similar in size to that of a virus. That means smaller than the bacterial cell, and they are known to cause various diseases even in humans also. Okay, what are the examples of the disease caused by the prions? Example: the scrapie disease in the sheep or the goat. Okay, bovine spongiform encephalopathy, BSE, in the cattle. It is also known as mad cow disease. Okay, then feline spongiform encephalopathy in the cats, Creutzfeldt-Jakob disease, Kuru disease, which is known as laughing death in case of humans. In case of humans. So these are some of the dangerous diseases which are caused by the prions. What are the prions? They are infectious particles which are protein in nature. They do not contain any nucleic acid. Okay. So this is prions. Now Let us see what NCRT has to say about these miscellaneous structures. Okay, we'll be having a quick reading of the NCRT, and we'll be seeing if there, and if if there is something new or something which we have not discussed is mentioned here. So, in the five kingdom classification of Whitaker, there is no mention of lichens and some cellular organisms like viruses, viroids, and prions. These are briefly introduced here. All of us who have suffered the ill effects of common cold or flu know what effect viruses can have on us. Even now, we have seen the lockdown in the entire world, and that too because of the virus. So now we know that what are the potential of the viruses, right? So even if we do not associate with our condition, viruses did not find a place in classification because they are not considered truly living. If we understand living as those organisms that have a cell structure, if cell structure is the criteria which we use to define a living organisms, and viruses are not truly living because they are not made up of cell, right? Now viruses are non-cellular organisms that are characterized by having an inert crystalline structure outside the living cell. Okay, inert crystalline structure outside the living cell. Now once they infect a cell, so outside the body they stay inert. Once they infect a cell, they take over the machinery of the host cell to replicate themselves. They start replicating themselves and they kill the host. Would you call viruses living or non living? So the answer is they are connecting link. They are present in between living and non living. The name virus, that means venom or poisonous fluid. Okay, the name virus means venom or poisonous fluid. Okay, was given by Dmitry Ivanovsky in 1892. Recognize certain microbes as the causal organisms of the mosaic disease of the tobacco. These were found to be smaller than bacteria because they pass through the bacteria proof filters. Now, Bajering demonstrated that the extract of the infected plants of tobacco could cause infection in healthy plants and called the fluid as contagium vivum fluidum, that means infectious living fluid. Then Stanley in 1935 showed that viruses could be crystallized, and crystals consist largely of proteins. That means viruses, they mainly consist of proteins. They are inert outside. They are specific host cells. That means they do have a specificity towards their host cells. And viruses are obligate parasites. They are dependent upon their parasites in an obligate manner. In addition to proteins, viruses also contains genetic material that could be either RNA or DNA. No virus contains both RNA and DNA. A virus is a nucleoprotein and the genetic material is infectious. In general, viruses that infect plants have single stranded RNA, I told you, and viruses that infect animals have either single or double stranded RNA or double stranded DNA, but mainly it is double stranded DNA. Bacterial viruses or bacteriophages are usually double stranded DNA viruses. This is also an important line. The protein code called capsid is made up of a small subunit called capsomeres and this capsid protects the nucleic acid. These capsomeres are arranged in helical or polyhedral geometric forms. Viruses cause disease like mumps, smallpox, herpes, and influenza. AIDS in humans is also caused by a virus. In plants, the symptoms can be mosaic formation, leaf rolling, curling, yellowing and vein clearing, dwarfing, or a stunted growth. So these are some of the features of the disease which is caused by viruses in plants. Okay, these are some of the features like the mosaic formation, patches formation, leaf rolling and curling, yellowing and vein clearing. Okay, dwarfing and stunted growth. These are some of the features or characters of the plant which is affected by virus. Okay, so this is the image. See, this is the image of tobacco mosaic virus, and this is the image of bacteriophage. We have already seen the same structure, right? Now, talking about the viroids, prions, lichens. So, viroids in 1971, Theo Diner discovered a new infectious agent that was smaller than viruses and caused potato spindle tuber disease. It was found to be a free RNA. It lacked the protein code that is found in viruses, hence, the name viroid is given. This is how they differ from the virus. They do not contain a protein code. The RNA of the viroid was of low molecular weight. This is also another important point. Now, talking about the prions, in modern medicine, certain infectious neurological diseases were found to be transmitted by an agent consisting of abnormally folded proteins. So, what are prions? These are abnormally folded proteins. The agent was similar in size to viruses. These agents were called prions. The most notable disease caused by prions are the bovine spongiform encephalopathy, commonly called as the mad cow disease in cattle, and its analogous variant, Creutzfeldt-Jakob disease in humans. Okay. It is also known to cause kuru, that is laughing death in humans. Now, talking about the lichens. So, lichens are symbiotic association, that is mutually useful associations between the algae and the fungi. The algal component is known as the phycobiont and the fungal component is known as the mycobiont, which are autotrophic and heterotrophic, respectively. Algae prepare food for fungi and fungi provide shelter and absorb mineral nutrients and water for its partner. So close is their association that if one saw a lichen in nature, one would never imagine that they had two different organisms within them. That is, the structure does not seem like this is the algal part or this is the fungal part. Okay. Now, lichens are very good pollution indicators. They do not grow in polluted area. I told you it is sensitive to sulfur pollution. Right. So this was about the NCRT session. So guys, we have now completed the entire unit till the plant kingdom part. I have not covered the animal kingdom though. Okay. So this was the part which was left in that particular chapter. So now it is over. So before taking your leave, let me again tell you my timings. I'm live daily at 5 p.m. on your favorite channel, which is Let's Crack NEET UG in English, where we are continuously providing you live lectures of all the three subjects, physics, chemistry and biology, so that you can study well and you can study conceptually and you secure a really good all India rank in NEET UG examination, because your target is NEET UG and we also want you to become a doctor. So with our combined efforts, we can definitely make our dream come true. Okay. Apart from this 5 p.m. session, I'm also live at 9 p.m. with my practice session. Every day, I bring a practice session of different chapters where we, for at least 1 hour we practice questions. Okay. And these are really important sessions. So I advise you all to attend these sessions, regularly. Then I'm also live at 11 a.m. on Unacademy Learning app for my free of cost special classes, guys. The link for my Unacademy Learning app profile is provided in the description box. You can follow me there to get all the updates regarding my class timings. And again, if you have still not subscribed for Unacademy Plus or Unacademy Iconic subscription, so please do not delay and do not waste your time. Go buy your subscription and do not forget to use my referral code AG001 to get an instant 10% discount and get a chance to enroll in my personal batch. Okay, guys? So, see you again tomorrow. Have a nice day. Thank you.